welcome to today's webinar on 7 critical steps for optimizing your lateral flow assay. In this webinar, we will discuss several key components that are often overlooked when optimizing a lateral flow assay, including tips to maximize your results. I'd like to really thank you for joining us. My name is Rebecca Hart, and I work here at nanoComposix in the Sales and Marketing Department, and I will be moderating today's webinar. I would now like to introduce Dr. Steven Olderberg, founder and CEO. Steve?
Thanks, Rebecca. So while we're waiting for a few more participants to join the webinar, I can just give you a brief overview of nanoComposix. For over 10 years, we've been a leader in the manufacturing of precisely engineered and highly characterized nanomaterials. We have over 500 different sizes and shapes and surfaces that can be produced at scale under a GMP and ISO 1345 compliant quality system. But increasingly, our role is not only to produce the materials, but to help people utilize those materials to make innovative and high-impact products. So no matter where you are on this road to commercialization, we can help. And for the early stage R&D, our customers use our metal and metal oxide nanomaterials as building blocks to rapidly prototype and test concepts. And then often we'll have people come back and ask for larger quantities or custom formulations of the materials. And then we'll be involved in helping them integrate those particles into the final product. We have over 20 different active collaborations and supply agreements with really a wide range of different customers, government agencies, large companies, and startups. Out of the startups, a few to note, there's a company called Sienna Biopharmaceuticals. It's a dermatology company that's using some of our silver nanoplates as a photothermal treatment for acne and light hair, laser hair removal. There's a Color Security. It's a color changing inks for the security industry. We're working with a company called Optiglow that's developing one-way illumination films for advertising on windows. Viveware is an antimicrobial compression sock for foot ulcers. And we've got a whole number of companies that are doing sensor applications. And then finally, one of the areas that we're most excited about is lateral flow. Combining our new ultra-bright reporter particles with covalent chemistry and mobile phone-based reader technologies, we really feel that this is going to be just an explosion of use, and as individuals take charge of the monitoring of the critical biomarkers that provide them with this unprecedented level of detail on the current state of their health. So we've been helping customers develop these assays in a range of different areas: stress markers, heart attack prediction, pregnancy risk, harmful chemical exposure, amongst others. So with that, I'd like to introduce Jean and Rhea, who are going to be giving a detailed presentation on some of the critical elements of lateral flow development.
Thank you, Steve and Rebecca. I'm Jean. And I'm Rhea. During this webinar, we'll give a brief overview of some of the key optimizations to perform to maximize the potential of your lateral flow assay.
Hi everyone, thank you for listening in today. In today's webinar, we're going to discuss 7 critical steps for lateral flow assay optimization. There are a ton of additional steps for development that need to be considered, but we want to break down just 7 of these steps to really provide the backbone of lateral flow development. Today we will be discussing the selection and optimization of the particle, the antibodies and affinity reagents, conjugation techniques, the conjugate pad, sample pad, and running buffer and sample handling. We hope that by discussing some of the problems that can be encountered and the techniques that we've used to solve these issues, we can help make your assay development successful. By the end of this webinar, our goal is to provide you with some of the basic tools for assay development and to convince you of a few concepts that are the theme to making your assay successful. The first is that consistency in development is critical. There are so many inconsistencies that you'll encounter in lateral flow that it's really important to keep things consistent with your protocols and limit variation as much as possible. The second is that optimization is assay dependent. This means that what works for one assay won't necessarily work for another assay. You can build more tools to use, but almost everything needs to be screened empirically for each particular test. Lastly, the process is iterative. We're going to discuss 7 of the steps required for assay development, but as you go through these steps, you often have to revisit some of the earlier parameters that you have previously chosen. So now we'll get started with an overview of the basic lateral flow test strip components and move on to breaking down each of the 7 steps that we've outlined.
Lateral flow assays are simple, inexpensive devices used to detect the presence or absence of analyte in a sample. They're intended for point-of-care use, delivering test results within 30 minutes of sample collection. They can be used in clinics and in the field without expensive or bulky equipment. Each assay is made of just a few simple components: a sample pad to which the sample is applied, a conjugate pad which contains the dried conjugate, a membrane striped with a test and control line, and a wick pad which absorbs excess sample and aids in capillary flow of the sample and conjugate through the membrane. All test components can be held in place with a compatible adhesive to a backing card, and often each test is housed in a cassette so that the end user only sees the membrane and the test readout. While the components of lateral flow assays appear to be minimal and straightforward, the chemistry involved and the optimization processes that ensure all pieces work in harmony can be complicated. The remainder of this presentation will briefly touch on several optimizations that are required to make the most of your lateral flow assay. One critical assay component is the particle or probe, which is conjugated to the detector antibody. It's the probe that gives rise to the colored response of the test and control lines. When choosing a particle, one of the first things to consider is what type of affinity reagent you'll be conjugating. Passive conjugation methods work well for conjugation to whole antibodies, whereas covalent conjugation methods can be used for conjugation to whole antibodies, antibody fragments, and small molecules. nanoComposix's bio-ready nanoparticles for covalent conjugation have a carboxylic acid on the surface, allowing for use with EDC and sulfo-NHS chemistry to form amide bonds with primary amines on the antibody. Because the number of amines and their physical position on the antibody are antibody dependent, empirical testing is required for each antibody. In some cases, antibodies that simply don't work for passive conjugation methods can work particularly well for covalent conjugation, and vice versa. Another consideration for particle selection is assay sensitivity. If an assay doesn't reach the clinically relevant range, there's simply no commercial market for it. At nanoComposix, we have developed a line of bio-ready nanoparticles that are brighter and more robust in comparison to standard 40 nanometer gold. Later in this presentation, we'll cover alternatives to 40 nanometer gold that can lead to an increase in assay sensitivity. Sample requirements are another factor to consider when choosing whether covalent or passive conjugation method is right for your assay. Covalent conjugates are significantly more stable in high salt environments, and the antibodies won't desorb under high protein or surfactant conditions. If your assay requires the use of harsh extraction buffers, high salinity, or harsh detergent formulations, covalent conjugation is the way to go. The type of assay you are building is another consideration for particle selection. We almost always find that covalent attachment is the preferred method when building a competitive format assay. This is because it's easier to modulate the number of antibodies per particle while maintaining colloidal integrity and stability, which can be greatly beneficial in this format. When building a quantitative assay, conjugation conditions are easy to control and reproduce using covalent methods. It's also critical that the probe itself be reproducible from lot to lot. Another thing to keep in mind during particle selection is cost. It's important to understand the market your assay will be sold into and the price point of the finished product. nanoComposix's line of bio-ready nanoparticles are competitively priced and come with the added value of extensive characterization, tight specifications, excellent lot-to-lot reproducibility, as well as unmatched customer support. On this slide, we've prepared samples of 40 nanometer gold and our bio-ready gold nanoshells at the same particle count. The color of the gold nanoshells is significantly darker than the 40 nanometer gold. When using conjugate probes with a higher per particle optical density, fewer binding events at the test line are required to visualize a test result, which is why we regularly see an increase in sensitivity with the gold nanoshells. On the following slides, we will show some case studies using solid gold spheres, which appear red to the eye, as well as some of our gold nanoshells, which appear as a blue-green color.
The next topic that we're going to cover is antibody selection. One of the most critical factors in developing a lateral flow assay is choosing the best antibodies for your particular test. The ultimate performance of the assay is dependent on the kinetics, affinity, and steric properties of the antibodies used in the system. The antibodies are really the workhorse that make the whole system work. No matter how much optimization you do, it really boils down to the antibody performance. All of the development steps are being done to give your antibodies the best conditions to maximize their performance. This is the main reason why optimization is assay dependent. Because the assay depends on the antibody, and every antibody has unique properties, they will require different conditions to be the most efficient. There are multiple types of lateral flow formats, and two of the main formats are shown here. For both formats, the schematic is showing what would happen at the test line with a positive or negative sample. The first one is a sandwich assay. This uses an antibody bound to a nanoparticle and an antibody bound to the test line to form a sandwich with an analyte and generate a positive signal. With a negative sample, no sandwich is formed and therefore no signal. The second type here is a competitive format. This is used when an analyte only has one epitope for antibody binding and can't bind two antibodies simultaneously to form a sandwich. In this case, the analyte can be bound to the membrane with a carrier protein, and it competes with the analyte in a sample to bind the antibody conjugated to the particle. This results in a decreased signal for a positive test and an increased signal at the test line for a negative test. The signal is inversely proportional to the analyte concentration. When selecting antibodies, there are many factors to consider, such as availability. Are your antibodies commercially available, or will you need to get them custom made? Will you use monoclonal or polyclonal antibodies, or a combination of both? Specificity in terms of where on the analyte is your antibody binding. If you're forming a sandwich assay, you want to make sure that the antibodies can sterically bind to two different epitopes on the analyte. You need to look at cross-reactivity. You can carefully read any supplier information about cross-reactivity and purity. Cost and supply can also be an important thing to consider at the early stages of development before investing a lot of time and resources. Affinity, which really needs to be tested empirically to determine if your antibody has a high enough affinity to bind the analyte in the lateral flow format. And lastly, here is stability. Some antibodies are more stable than others, which make them easier to work with for development. Overall, the antibodies need to be screened empirically in the lateral flow format. Even if there was a pair that worked best in an ELISA, that doesn't necessarily mean that they will be the best pair for lateral flow. Although the assays are similar in theory, the lateral flow assay relies on the antibodies to bind quickly and efficiently to the analyte with an extremely short contact time in comparison to an ELISA. Because of all of the factors that are involved with antibody selection and the importance of choosing the right pair, we like to screen as many antibodies as possible in a matrix of capture and detector antibodies. We pair each one of the antibodies as the antibody conjugated to the particles and the other striped as the test line reagent. Using covalent conjugation can really help with this process because there's no need for pH titrations. This table here shows a matrix of 10 different antibodies and which ones perform the best. The antibodies were initially down-selected from a much larger number based on clone, epitope specificity, availability, and purity. On the table shown here, the number corresponding to a given pair represents the signal intensity with a positive sample. All of the pairs were also run with negative samples to ensure that the signal isn't a result of nonspecific binding. The boxes in light blue indicate a fairly decent signal, while the boxes in dark blue indicate the strongest signal seen. In the image to the right of the table, you can see that the left strip did not pair and showed no positive signal, while the middle strip shows a medium positive signal, and the strip to the right shows the strongest signal. This was the antibody pair that was chosen for further optimization in this case. The next topic that we're going to cover is the nitrocellulose membrane. The membrane is the part of the assay that has your test and control line reagents. It plays a huge role in the speed and performance of the assay, while also provides the visual readout to the end user. The image to the left shows an SEM image of the membrane, which is made of mesh-like nitrocellulose fibers. The image on the right is an example of one type of machine used to dispense the test and control lines onto the nitrocellulose. This particular one is an isoflow reagent dispenser, which uses contact dispensing to stripe the lines. Here we have outlined some of the main areas to consider when choosing the correct membrane. First of all, you need to consider the various manufacturers of membranes that are available. Manufacturers treat their membranes with proprietary components that make them hydrophilic and able to bind to protein. This gives them different properties and makes them more suitable than others for particular assays. So again, these need to be screened empirically for every test. What worked well for one assay might not work well for another. The next factor is the speed and type of assay. Some manufacturers have backed and unbacked membranes, which can affect the performance of the test. The other main factor is the pore size and capillary flow rate. The capillary flow rate is the amount of time it takes for water to wick 4 centimeters through the membrane. Membranes with a smaller pore size are slower and can usually provide better sensitivity because they allow a greater time for reaction between the reagents as they flow. On the other hand, faster membranes are often less sensitive but tend to work better for competitive assay formats, for assays that have nonspecific binding issues, or for assays that need a faster read time, such as 5 minutes instead of 20. The last aspect here is the striping parameters. These can include the concentration of your test and control line, the dilution buffer used, the dispense rate and speed, the type of dispensing used, such as contact or non-contact, and the methods used for drying and curing your membrane after it's been striped. After you stripe your membrane, it's really important to fully dry it in a forced air convection oven at 37 degrees Celsius and store it in a desiccated environment. If you don't have a dry room where the humidity is less than 20%, store all of your materials in a sealed foil pouch with desiccant or some other method to ensure less than 20% humidity for the materials. This will help with consistency of your results. In this slide, we put an example of a competitive assay that needed some optimization of the membrane striping parameters. The control line looked good with a clear line at about 1 millimeter width, but the test line was blurry and spread out. This issue at the test line is common when you're striping analyte bound to carrier protein, often used in competitive assays, such as THC-BSA conjugate for drugs of abuse. This is also common for large pore membranes, which is usually used for the competitive assay formats as well. So how do you fix this issue? The first thing we would address is the dispense rate and speed. A typical dispense rate for a medium speed membrane would be about 1 microliter per centimeter. For this assay, decreasing the dispense rate to 0.5 microliters per centimeter improved the line width and clarity. Since you are decreasing the amount of reagent being dispensed, you can also increase the concentration of the test line reagent so that you are putting equivalent reagent down at the line. The other potential issues are nonspecific binding, a strong leading edge, and ghost lines, which can be improved by adjusting the test and control line concentrations and adjusting your dilution buffer of the reagents. The dilution buffer can be adjusted by changing the salt content, buffer molarity, and addition of small concentrations of additives to stabilize your protein.
The next topic we'll cover today is conjugate optimization. In this presentation, we refer to a conjugate as a nanoparticle probe that is bound passively or covalently to a detector antibody. There are several steps that can be individually optimized during antibody conjugation for both passive and covalent methods, including optimizations for the antibody purification buffer, the conjugation pH, the antibody to gold ratio, antibody incubation time, blocking steps, and conjugate diluent formulation. This slide shows 2 strips run with positive samples where we expect to see a signal at the test line. The strip on the left resulted in acceptable results. However, switching the antibody purification buffer from 10 millimolar phosphate to 1x PBS resulted in a stronger signal intensity at the test line. One of the first things you can optimize is the antibody purification buffer. Passive conjugation is very sensitive to salts, and having too much salt present will result in colloidal instability. For passive conjugation, potassium phosphate and borate buffers are often used. For covalent conjugations, many buffer formulations are compatible so long as they don't contain primary amines. Notably, tris buffer or anything with sodium azide as a preservative are not compatible with EDC sulfo NHS conjugation. Antibodies provided with interfering substances can easily be purified with spin filters or by dialysis. Always refer to the antibody certificate of analysis to help select buffer formulations and pH and to ensure proper storage and handling of your antibody. For both passive and covalent methods, the ratio of antibody to nanoparticle should be tested empirically. For passive conjugations, you are really limited to the antibody loading that results in colloidal stability. The strips you see here were prepared with particles at the same volume and OD with varying amounts of antibody. Each test was run with the same positive sample. In this screen, 20 micrograms per 20 OD mil antibody loading gave rise to the strongest positive signal intensity, while higher and lower antibody loadings both decreased sensitivity. In many cases, Suboptimal antibody loading can also lead to nonspecific binding at the test line. Another important optimization to perform is the conjugate diluent formulation. The conjugate diluent is the buffer solution the conjugate is stored in prior to dispensing and drying on the conjugate pad. It often contains a buffer, proteins, polymers, surfactants, and sugars to help stabilize the conjugate upon drying, to aid in long-term stability, and to allow for good resuspension and release of the conjugate when the sample is applied. On this slide, we show 3 positive test strips and expect to see a strong signal at the test line for each strip. Each was run with antigen spiked into freshly collected human saliva. Typically, when screening conjugate diluents, we begin our screen with a straight formulation of PBS with a small percentage of BSA. We then screen detergents, proteins, and polymers individually and at varying concentrations in an effort to decrease background and nonspecific binding, promote consistent conjugate release and flow, and to increase the test line intensity with positive samples. In this example, several proteins and detergents were screened, with 0.25% casein as well as 2% tween 20 giving rise to optimal conditions. In addition to diluent components, screening the pH of the conjugate diluent may improve assay results, especially for larger particle conjugates. It's important to note that to reach these results, each component listed here, along with many others, were examined individually and at varying concentrations. The most promising modifications were combined to achieve the results presented on the right. Unfortunately, there is no magic formula that works for every assay, and there's a lot of empirical testing needed to achieve the best results. On this slide, we'll go over some other optimizations for the conjugate diluent. Here we present 2 strips that were both run with a negative sample where we expect to see no signal at the test line. Initial testing of the antibody pairs showed significant nonspecific binding at the test line, as shown in the image on the left. With the addition of blocking agents, specifically BSA and casein in this case, and also the addition of tween 20, the nonspecific binding at the test line was eliminated while maintaining a positive signal at the test line with a positive sample. Other optimizations to perform for the conjugate include pH optimizations, antibody incubation time, and blocking steps. For passive conjugation, the isoelectric point of each antibody is different, so the pH of the colloidal solution should be tested empirically to determine which pH works best for each antibody. For covalent conjugation, the activation of carboxyl nanoparticles with EDC and sulfo-NHS is best carried out between pH 5 and 6. Our carboxyl bioready nanoparticle solutions will self-adjust to this optimal pH upon the addition of reagents following our protocols. After activation, we centrifuge our particles and resuspend the activated nanoparticle pellet into a reaction buffer before adding the antibody. The reaction of sulfo-NHS activated nanoparticles with primary amines is best performed at pH 7 to 7.5. NH esters will hydrolyze within hours or minutes depending on water content and the pH of the reaction solution. They have a half-life of 4 to 5 hours at pH 7, 1 hour at pH 8, and only 10 minutes at pH 8.6. We recommend carrying out the conjugation to antibodies between pH 7 and 7.5 for consistency. The antibody incubation time will differ from antibody to antibody, especially for covalent conjugation methods. The number of primary amines and their physical location on the antibody is antibody dependent, making the reaction kinetics different for each antibody. Typical antibody incubation times for covalent conjugation range from 5 minutes to an hour. Including a blocking step can be crucial for some conjugates while having little impact for others. The purpose of blocking is to increase particle stability and decrease nonspecific binding. There are a number of commercially available reagents for blocking. The next optimization we'll discuss is the conjugate pad. The purpose of the conjugate pad is to absorb the conjugate fully and evenly while it's being dispensed so that each test delivers the same number of conjugated particles. It should maintain the integrity of the conjugate upon drying and also during long-term storage. and aid in the release of the conjugate upon wetting with the sample media. Conjugate pads are most often made of glass fiber, cellulose, or other synthetic materials. Pretreatments can be employed to help particle release and decrease nonspecific binding. An important consideration when choosing a conjugate pad is the volume you are planning to dispense per test. If your assay requires a large volume of conjugate, you may need a conjugate pad with a high absorption. If you need an ultra-quick test readout, a less dense conjugate pad will likely perform better. After dispensing the conjugate pad should be dried in a forced air oven at 37 degrees and then stored in a desiccated environment until it is ready to be used. We're now going to take a quick break while we ask our listeners to take a poll. For those in the audience, do you feel more comfortable using passive or covalent conjugation methods? In our hands, we see a lot of benefits to using covalent conjugation. That said, even with optimizations, we occasionally see that for some antibodies, the passive conjugation gives rise to better results.
So it looks like most of you guys are more comfortable with covalent than passive, which is interesting and I think bodes well for having extra control over your antibodies and your conjugates that you're producing.
The next topic that we're going to talk about is the sample pad. The sample pad is the first point of contact between the sample and the test strip, and it's critical for delivering the sample of interest to the reagents in the strip. This slide shows a small subset of the various types of fluid that can be used on a lateral flow assay. The biological fluids here are shown in a gradient of the difficulty of sample collection, starting with CSF to venous blood collection, fingerstick blood, urine, and saliva. More and more focus is being moved towards saliva as a biological fluid for assays because of the ease of collection. There are a wide range of other samples that aren't pictured here, including insects, stool, water supply, and soil samples, among many others. For a lateral flow assay, the sample pad needs to be able to filter out unwanted portions of the sample, such as red blood cells. It needs to neutralize the sample, such as the urine pH, and it needs to normalize the sample, such as the viscosity variations seen in saliva samples. Most importantly, it needs to do all of these functions rapidly and consistently before wicking the sample through the test strip. It needs to provide the same recovery of Analyte regardless of the sample variation. There are many materials available to be used as sample pads, and they need to be screened for each assay. They are usually made of glass fiber, cellulose, or synthetic material. There are also many treatments that can be applied to the material to help with some of the critical functions of the sample pad. Similar to the conjugate pad, these can include adding blocking agents, polymers, or surfactants. Sample pad materials can be treated by immersion into the buffer or dispensing the buffer mechanically. The treated pad should be fully dried in a forced air oven and stored in a desiccated environment with less than 20% humidity. One important factor to consider when screening sample pads is Analyte compatibility. This slide shows two test strips that are identical in every way except the sample pad material. The strips were run with a positive sample, so a relatively strong signal is expected at the test line. As you can see in the second strip here, the test line intensity is significantly decreased. Since the sample pad is the only component that's different from the first strip, it shows that the material used for this isn't optimal for this assay, and it's most likely binding the analyte and not allowing it to flow through the membrane. The main point here is that if you performed all of your optimization with sample pad number 2, you will never see the full potential of your assay system. Another important point is that sample pad number 2 could be optimal for a different test, which makes it important to screen for each individual assay. Another important factor is to optimize for the particular sample medium that you're using. In this slide, we're running test strips with saliva. The assay was optimized and working great with spiked buffer samples, which we will discuss later, in the range that we needed to detect. After a huge amount of optimization, a real saliva sample was run, and there was absolutely no flow through the test strip, as you can see in the test strip all the way to the left for assay number 1. This is a common problem when switching from buffer to real samples. To improve this, we screened a wide range of sample pad materials with a wide range of saliva donors. The next strip shows the improvement made by changing from a glass fiber material to a cellulose material. However, you can also see that the background was still really high and that the flow was not perfect. By treating the sample pad with a high concentration of salt, we were able to improve the flow and get valid results at the test line. The assay number 2 on the right was another assay that also used saliva as the sample medium, using gold nanoshells as the detector particle. We tried to apply this same sample pad material and treatment shown on the strips to the left, and the flow is extremely poor, as you can see in the first strip for this assay. This required an increase in the pH, salt, and surfactant used in the sample pad treatment to allow flow of the saliva through the strip. Because of the high pH and high concentrations of salt and surfactant, it's unlikely that passive conjugates would stay stable under these conditions. By using covalent conjugations, we can use these buffers to improve treatment of the sample without affecting the assay results. This next slide here shows a sample pad evaluation using whole blood as the sample medium. The original prototypes were made with just a standard glass fiber when testing buffer spiked with antigen. However, the test needed to be run using finger stick whole blood, so the sample needed to retain all of the red blood cells while allowing the serum and the analyte to flow through the test strip. For all of these strips shown here, the blood was negative for the analyte, so we were looking for retention of red blood cells in the sample pad, and we were looking for a strong control line on the membrane. For this particular assay, we screened 10 different blood filter materials, 5 of which were not compatible with the analyte because the signal at the test line was lost when using spiked buffer. Many of the red blood cell filters also had high leakage of blood cells through the test strip, and some even caused extremely high nonspecific binding, which may be a result of hemolysis or other factors. Using an improved running buffer, we were able to improve the results with some of these blood filter pads, as you can see in strip B. There was still some leakage of the red blood cells onto the membrane, so the test strip width was increased in order to increase the surface area of the sample pad and retain more blood cells. So now we're going to move on to the last topic. For this topic, we're going to discuss the running buffer and sample preparation used in assay development. Like many of these topics, there could be a whole webinar that just addresses this topic alone, so we will try to touch on some of the most important points here. A running buffer might be used in some assays and might not be required for others. For example, in some saliva or urine assays, you may not be diluting your sample. In this case, you would not have a running buffer. But for other samples, such as whole blood, you will only be applying a small volume, and it'll need a running buffer to chase the sample. In the cases of solids, such as insects or soil samples, you will need a buffer to dilute and extract your sample to apply to the test strip. By optimizing the running buffer, you can improve the flow, eliminate nonspecific binding, and improve the binding of your analyte. The running buffer can include a buffering component, such as PBS, polymers, surfactants, and proteins. When you're dealing with samples, it's really important to keep in mind that all of the other endogenous components of the sample may interfere with binding to your analyte. This can be mitigated by optimizing the running buffer or with some of the other optimizations listed in the sections above. The other issue to think about is the stability of the sample. Not only is the stability of the analyte important, but the stability of the rest of the components of the sample are important as well. So how do you know what type of sample to start testing? Once again, it depends on the type of assay you're building and what reagents are available for use. If you're able to obtain purified antigen, this flowchart might be the type of system you would use. For initial development, you can spike purified antigen into buffer to give you an idea of the performance of your antibodies. This can tell you if you're in the right range and if your antibodies are functional. You can then transition into spiking purified antigen into the real sample medium. This can be difficult in some cases where you're looking for something that is endogenous to human samples, so it can be hard to find a negative. But if you can include this step, it'll help you optimize for the specific sample type, such as urine or saliva. Lastly, it's really important to transition to real samples early in development. 99.9% of the time, this transition is going to set you back, so it's best to perform optimizations with real samples, since you may have to revisit a lot of the earlier steps of optimization to make it work. An example flowchart here is for the development of a standard pregnancy test to detect HCG. Early in development, you can purchase purified HCG and spike it into buffer samples. This will help guide your antibody selection, and it'll let you know if you're in the right range to detect pregnancy. Since HCG is usually not an endogenous hormone if you're not pregnant, you can transition into spiking HCG at known concentrations into negative pooled urine. At this stage, you can perform many optimizations to make the assay work with this type of sample. Finally, you can transition into different urine samples that are really positive and negative for HCG and make sure that your system is compatible with all of the variability that can occur between samples. At this point, it's really important to make sure that the HCG concentrations are verified by an outside method. This slide here is to show an example of the dramatic effect that the running buffer can have on your assay performance. For assay number 1, 2 different buffers were used for negative samples, so we expected to see no signal at the test line. As you can see in the first strip to the left, buffer 1 caused a huge increase in nonspecific binding at the test line. Obviously, this buffer wasn't chosen for further optimization. However, for assay 2, both buffers looked relatively equivalent and neither caused an increase in nonspecific binding. This is just another example to show that what works best for one assay might not work for another. So sample stability is important because biological samples change over time. This is an example of an assay at the early stages of development where the analyte was spiked into buffer. For some analytes, you can dilute and store in the fridge or frozen for extended periods of time with no change to your analyte. However, this is an example where even storage overnight caused the sample to become unstable and caused a loss of signal that you can see on the strip to the right. It's important to track the stability over time because it may be that your assay is not giving you the best sensitivity due to your analyte and that you're not seeing the peak performance of your system. Another aspect to consider is in the stability of the sample medium and not just the analyte itself. Regardless of analyte stability, other components in the sample, such as serum, blood, or saliva, can break down over time and affect the results. For example, mucins and proteins can break down in saliva over time or with freeze-thaw cycles, which could actually help improve the flow and binding of the analyte. This might make your assay look great, but once you start running fresh samples, you'll realize that there's a lot more optimization required to make the system work with fresh saliva.
So those were the 7 optimizations we had time to cover in this webinar. I'll now briefly touch on some other considerations to keep in mind. Lateral flow tests should have a shelf life of 12 to 24 months when stored at ambient temperature and humidity. Sample preparation and ease of use are critical. Many lateral flow tests will be run by people with limited training, so it's important that the assay and protocol be as robust as possible. The wick pad can significantly impact assay performance and runtime and should be tested empirically for each assay. A cassette is another feature that is useful for many lateral flow assays. The cassette can ensure that all the test components overlap with the correct contact pressure and aid in better flow and test reproducibility, among other things. We touched briefly on quantitative versus qualitative tests when discussing particle selection. However, it's important enough to reiterate that this should be taken into consideration early in assay development. For qualitative tests, and especially quantitative tests, a reader may be a desirable assay component. As we mentioned earlier in this presentation, this is a brief overview of several of the optimizations, and many topics we covered today could be their own hour-long webinar.
Our goal for this webinar was to give you some basic tools and techniques for lateral flow assay development. Hopefully by walking through these 7 steps, it also helped to prove the importance of some of these take-home messages so that you can make your own assay development successful. First is that consistency is key. During development, small changes in your protocols can cause an extreme variation in your results, which will make it difficult to optimize and improve your assay. Secondly, optimizations are assay dependent. What works for one assay may not transition and work for another. It's important to empirically test reagents, materials, and conditions for each individual test. Lastly, the process is iterative. We've listed 7 steps here, but what looks like 7 steps actually turns into a huge matrix of testing. You might choose your antibodies, particles, and membranes, only to realize that some of these parameters are not optimal after you change some of your conjugate conditions. It's important to go back in the process once you change certain parameters and make sure you're not losing performance based on previous results. Thank you for listening to today's webinar. At this point, we're going to pass you back to Rebecca. She's going to open up the webinar for questions that Jean and I can answer for you. If you're looking for more information, we also have a lateral flow assay development guide and multiple white papers that are available on our website for your reference.
Thank you, Jean and Rhea, for that valuable information. Some of our listeners have submitted questions, so we're going to go over some of those. If you still haven't you'd like to ask a question and you haven't yet, please feel free to use the question pane. Just type in your answer. As we go through the questions, you'll have the opportunity for us to answer those. So our first question comes from Andrew today. For a competitive assay format, what conditions do you recommend for conjugation?
For a competitive assay, we highly recommend covalent binding for the conjugate. For competitive formats, it's really important to limit the amount of antibodies per particle because that can actually improve your sensitivity. By decreasing the amount of antibody and incubation time, it can cause regular passive conjugates to be unstable. So by using covalent conjugates, you have a lot tighter control over that.
Okay, next question comes from Keith. He asked, what type of conjugation do you recommend for a sandwich assay?
That's really antibody dependent. We typically start by performing covalent conjugations, and if the end result doesn't look as promising as we think it should, sometimes we'll go back and try passive conjugation methods or select a different antibody to use.
Okay, we also have a question from Ben. Which particle gives rise to the blue signal on the test line?
So the nanoshells have a blue signal, and that's really due to the physics of plasmonics. So the reason why gold particles are used is that they have really unusual, gold and silver particles have really unusual optical properties at the nanoscale. So by changing the size of the shape, you can change their optical properties. And for 40 nanometer gold, it's a very efficient absorber in the blue and green regions of the spectrum, which makes the particles appear red. For the nanoshells, the absorbance is actually shifted off into the far red or the infrared, and that's why the particles appear blue. So it really just has to do with the physics of plasmonics, and the reason why we're using these materials is that they have very, very large cross sections, so that each binding event gives a very strong signal on that white nitrocellulose, giving you high contrast so you can get high sensitivity.
Thank you, Steve. This question is from Gabriel. When is a passive conjugation a good option? Do you have any examples of that?
It all comes down to empirically testing the antibodies. So depending on where the lysine residue on the antibody is, or how certain proteins fold and interact with while they're in solution, will depend on whether or not covalent conjugation method is practical for that antibody. In some cases, it's just important to test both methods empirically and see what gives better results. If some of the conditions we mentioned, like harsh extraction buffers or high salinity samples, are used in your assay, it may not be an option to use passive absorption, in which case, if you had an antibody that wasn't working well with covalent attachment, it may be worth looking into other antibodies.
Okay, thank you, Jean. This question comes from Keith. How do you control or monitor the stability of additives such as surfactants or salts on the lateral flow material?
There's a couple of ways to tell if your conjugate is losing stability. Often when the particles are aggregating together, you can visibly see a change in color. The optical density of the solution will drop, and if you have access to a UV vis, you can see an elevated baseline and a broadening of the UV vis spectra, which is indicative of particle aggregation. Another metric to measure that is by DLS, measuring the hydrodynamic diameter of the conjugate. But the quickest and easiest way to tell is simply by looking at them in solution.
Great, thank you, Jean. Our next question comes from Zoe. Is a competitive format better to detect lower concentrations of antigen than a sandwich assay format?
Not necessarily. It really depends what type of antigen you're looking for. Competitive formats are used when the antigen is usually really small, such as drugs of abuse or small steroid hormones. And it's used because the analyte itself can't bind to antibodies simultaneously. So it's not necessarily a sensitivity issue. It really depends on your analyte size and how many epitopes are available.
Thank you, Rhea. The next question comes from Gabriel. From your experience with particle types, size and shape, is there a most sensitive one for a given assay?
Yes, so that's a great question. That's a question we asked 3 years ago. Because we noticed that everybody was using solid gold spherical nanoparticles, and we had a library of 500 different sizes and shapes. And so what we did is we basically systematically went through our library of rods and star-shaped particles and different hollow particles and nanoshells. And at first pass, what we came up with was that these 150 nanometer gold nanoshells actually performed really well. And part of the reason is just the physics of that optical resonance. The silica shell or the silica core that we use in those particles is very uniform, so that there's a very low coefficient of variation in the size. And then also that silica core is less dense, and so these particles, even though they've got that same gold surface that works really well for lateral flow, it doesn't have the same density as a solid gold particle, so the flow characteristics are a little bit better. So as we found, that those nanoshells seem like they're doing a great job. Not to say that we might find something better, but right now that's where we're focusing our efforts for high sensitivity requirements.
Thank you, Steve. This question comes from Samba Shiva. Can we use other particles for color detection other than nanoparticles?
So there's a whole bunch of different probes in terms of fluorescent beads and dyed beads and upconverting phosphors that are all potential candidates. And in some cases, those have even higher sensitivities. One of the drawbacks is that you typically need a particular reader that's going to be coupled with that. So if you're looking for ultimate in sensitivity, there are other options. It's just that you're going to have a requirement of having a more complicated reader, which works fine for the lab, but in some of these more mobile or far forward applications where you want to use it from a consumer perspective, it's more convenient just to be able to look at it and see contrast.
Great, thank you. This question comes from Nadia. What blockers would you recommend to use for particle blocking after conjugation?
Again, that's something we empirically test for each assay. That can vary from proteins like BSA, casein. Occasionally we use fish skin, gelatin, and there are some other synthetic blocking agents that we found to be a little more reproducible from lot to lot from the manufacturer. There are a whole slew of blocking agents that are out there commercially available. So I typically would recommend screening as many as you can while also considering the cost.
This question comes from Liz. For screening purposes, do you usually start with passive by default and then move to covalent assay later?
We almost always start by screening with covalent conjugations. It makes the process a lot easier because you don't have to, it's not dependent on each antibody's isoelectric point. So for passive absorption, you have to screen which pH is best for conjugation. Versus covalent conjugations, you don't need to do that. So in one conjugation, you can conjugate a lot of different antibodies, and it makes it a lot easier to screen.
Okay, thank you. I really appreciate that. This question comes from Shima. What could be the best blocking agent for nitrocellulose membrane in the case of using coated gold with aptamers, the non-covalent binding or passive method?
I'm not sure if this is referring to blocking the actual nitrocellulose membrane. If it is, I would recommend using very low concentrations of proteins. We usually don't block the nitrocellulose membrane itself. That can cause some issues later in the manufacturing process. As far as other areas to block, I would recommend the same things that Jean mentioned earlier, such as proteins like BSA and casein. Surfactants can help, and also some different types of polymers like PVA and PEG.
This question comes from Tim. Is it better to pre-treat a sample pad by immersing or by direct dispensing?
Both methods work depending on your application. We like dispensing mechanically, either by hand or with a machine, because it's really important to control the amount of volume that's going on per sample pad. This can really help with reproducibility of your assay results, making sure that each test strip has the same amount of the components as the next test strip.
Okay, great. And I think that's all the questions that we have for now. I'd really like to thank you, Jean, Rhea, and Steve for sharing this valuable information to our audience. I'd like to thank everyone for attending today's webinar, the 7 critical steps for optimizing your lateral flow assay. If you have any further questions, you may reach our technical support staff at the info email address provided, listed on the screen there, or you can also contact us by phone at the number also provided there. And then on behalf of nanoComposix and our presenters, Jean and Rhea, I'd really like to thank you so much for joining us today.